Hello, my name is Ryan Schulze. I am the Director of Research and Development at Cytotheryx, and we are a preclinical cell therapy company based in Rochester, Minnesota. I'd like to thank the Lab Roots organizers for giving us a chance to go over some of our translational research efforts with you. And today I'm going to be discussing our development of a novel bioincubator for the large-scale production of high-quality primary human hepatocytes. So the problem that we're attempting to address is that of liver disease, and that's an issue that has resulted in significant morbidity and mortality in not only the United States, but also worldwide. And in this country, there are almost 5 million adults annually that are affected by various liver diseases. And these range from acute liver failure that is induced either by acetaminophen overdose or infection with hepatitis viruses to chronic conditions that are brought on, for example, by excessive and chronic intake of alcoholic beverages. Unfortunately, other than receipt of a liver transplant, there really are no currently approved therapies that are available for the treatment of liver failure. And therefore, there's this really significant and urgent unmet need for novel strategies to help these patients. And so, unsurprisingly, the scale of liver disease is coupled to this really incredible societal and economic cost, with liver disease, as you can see here, occupying a position within the top 10 most frequent causes of death in the United States, with something like 50,000 deaths per year. And additionally, the burden on patients should a liver transplant actually be required, and an organ that is made available is really extraordinary, with an average cost of something like approximately $800,000. So the question becomes, how can we begin to address the various causes of liver disease? And one of the paths forward would really be to have a large supply of very high quality liver cells on which we could, perform really much needed basic research, and then also two, to use them directly for numerous life-saving therapies. The problem is that there really isn't a large supply of high quality cells. This just doesn't exist right now, and the bottleneck is really a limited availability of human liver cells, otherwise known as hepatocytes, that are available for these purposes. And there's really a number of issues that are at play here. First and foremost, hepatocytes can't be grown in vitro, so that's outside of the body. Unlike pluripotent stem cells, for example, which can be expanded to really astronomical numbers in industrial bioreactors, the same just isn't true for hepatocytes. Now, the liver itself is remarkable in the sense that it has this really extraordinary regenerative capacity. And in fact, more than half of the liver can be surgically removed. And even then, hepatocytes will regenerate and the liver will very quickly grow back to its original size. But despite that fact, once those hepatocytes are removed from the liver, their capacity for regeneration and expansion essentially drops down to zero. And this is despite decades of really trying to coax them along. So therefore, as kind of a gold standard, much of basic science research, as well as toxicological and pharmacological studies, make a heavy use out of primary hepatocytes that are isolated from deceased donors and cryopreserved for on-demand use. But this brings up a second issue, and that's only that approximately about 9,000 livers become available each year that are really suitable for transplantation. And it's the remaining non-transplantable organs, many of which are suboptimal, that are being used for hepatocyte isolation and downstream liver disease research. So as a result, these primary hepatocytes, many of which have already been subjected to significant chronic injury, that's the material that's serving as the foundation for many of our existing research initiatives. And furthermore, there's a significant donor-to-donor variability of hepatocytes, which, as you can probably imagine, causes numerous problems in our ability to interpret scientific studies that make use of those cells. And so there's a number of alternatives to the use of primary hepatocytes that different groups have explored, including hepatoma-derived cell lines like HepG2s and Hep3Bs, HUH7s and HepRGs. And these are useful models, but despite their convenience, their ease of expansion, and their relatively low cost, they really lack the capacity to perform the full repertoire of functions that are performed by primary hepatocytes. So as you can see on this slide, hepatocytes really are these incredibly important and complex cells that play an integral role in host metabolism. And so hepatocytes, as you can see in this figure, they line the sinusoids of the liver. They're exposed to a mixture of both arterial and venous blood supply that comes from the hepatic artery here and the hepatic portal vein, respectively. These then drain eventually into the central veins and then out of the liver via the hepatic veins to the IVC. Hepatocytes make up about 80% of the mass of the liver. And they're essentially these protein secretory machines that produce the bulk of just about all proteins that are present in the blood supply. And I've just listed a few examples here, but these range from carrier proteins such as albumin and alpha-fetoprotein, iron-binding proteins such as transferrin, proteolytic factors like plasminogen. You've got protease inhibitors such as alpha-1 antitrypsin, and then numerous acute phase proteins, and then really a host of blood clotting factors. Hepatocytes also really play critical roles in processes such as lipid and carbohydrate synthesis and storage, secretion of bile precursors. The bile ultimately aids in the digestion of food, and then also the uptake of various important growth factors. And then finally, and most importantly, the hepatocytes are the central sites of drug metabolism. And this is where bioactivation and detoxification reactions occur. And they occur mostly in hepatocytes. So things like acetaminophen, alcohol, ammonia, these are all examples of compounds that really are dealt with by the hepatocyte. And it's the differences in hepatocyte to hepatocyte enzyme levels and function that really serves as the basis for our individual differences in responses to different drug treatments and variations in toxicity. So the use of these hepatocytes is really crucial in the development of new drugs in order to understand hepatotoxicity and possible physiological effects of treatments with new drugs. So because of the shortage of supply and lot-to-lot variability of primary hepatocytes originating from deceased donors, there exists a real need for large-scale preparations of not only a consistent supply, but also a clinically safe supply of primary hepatocytes. And these cells would serve like a hugely important function for small molecule research and toxicology studies, for example. And so here, hepatocytes are absolutely essential for the metabolic profiling that's associated with the test items that are being developed. And if it were possible to provide a really consistent source of cells with minimal lot-to-lot variability, that would really provide a significant advantage to the developmental workflow. Furthermore, there exists a great need for hepatocytes for basic research into liver-centric diseases such as the hepatitis viruses, HBV, and malaria in the basic research involved in those diseases. As I mentioned earlier, most of the cells that are currently available, most of the hepatocytes that are currently available are derived from tissue that's obtained from non-transplantable organs. And so in some cases, these organs are procured from individuals with communicable liver diseases. So it would really be advantageous to have a very large bank of hepatocytes that have been thoroughly vetted, not only for consistency, but also which have clinical safety profiles that would really enable us to use these cells for emerging life-saving technologies such as liver assist, as I've got here, or bioartificial liver systems, or for direct hepatocyte transplant into patients. And these are technologies that really are currently being developed as mechanisms to help bridge patients on the liver transplant waiting list, providing much needed support as they await on the list for being matched with a donor. And finally, the ability to produce hepatocytes at large scales lends itself to other exciting technologies in the gene therapy space, in which patients with rare metabolic liver diseases might benefit from having their own defective liver cells removed, corrected using advanced gene editing techniques, expanded to really large numbers, and then returned to that very same patient as a curative therapy. But this brings up once again, the biggest problem and challenge with these cells, and that's namely that they do not expand in vitro. So it's not as simple as being able to grow up hepatocytes to these really high densities and numbers that are needed for clinical therapies, just using a stirred tank bioreactor, for instance. So the question is, how can we get around this limitation of hepatocytes being unable to expand outside the body? So it turns out there's a very clever trick that can be used to regenerate hepatocytes in vivo, and it stems from research that was done on a critical enzyme that's involved in the metabolism of the amino acid tyrosine, and that's this enzyme called fumaryl acetoacetate hydrolase, or FAH for short. And so individuals that have genetic mutations in FAH that prevent its enzymatic activity have a disease known as hereditary tyrosinemia type 1, also known as HT1. And as you can see here in the bottom of this image where this dashed line is, FAH catalyzes a hydrolytic conversion of a hepatotoxic intermediate, so that's this fumaryl acetoacetate, into fumarate and acetoacetate. And when FAH is mutated, however, fumaryl acetoacetate begins to accumulate, and the patient with HT1 will eventually begin to develop really progressive liver and kidney dysfunction. So it's not curative, but these complications can effectively be prevented by treatment with a drug that's known as NTBC, which is actually inhibiting an enzyme further up the tyrosine metabolic pathway, HPD, here on this diagram, and therefore that prevents downstream accumulation of those toxic metabolites. So an understanding of this pathway is really important in that it provides supporting rationale for introducing healthy hepatocytes into an FAH deficient liver and then withdrawing NTBC support. And with time, the theory would be that these healthy cells, which have functional copies of FAH, would possess a selective growth advantage over the rest of the FAH null cells, and with time slowly begin to expand and repopulate the liver. And experiments have demonstrated that actually, in fact, is the case in multiple animal models. And importantly, it was shown at scale in the Science Translational medicine paper from 2016 that was led by a group at the Mayo Clinic, in which an FAH deficient pig was created and then engrafted with FAH positive hepatocytes. And as you can see in the immunohistochemistry at right, at a time point 2 months post-cell transplant and following periods of cycling the animal on and off NTBC, you begin to see these islands of FAH positive hepatocytes that begin to appear in this section taken from the liver, in which engrafted cells have started to expand within their surrounding environment of FAH null hepatocytes. And importantly, when the authors carried these animals out to 12 months of age, you can see that not only did the animals begin to thrive, as you can see in these growth charts at the top right of the screen here for pigs 2 and 3, but that it was possible to eventually remove them from NTBC therapy completely, as it's illustrated here by these gray vertical bars on the graphs, which represent time periods that NTBC was administered to the animals. So if you're looking at this by 150 days post-cell transplant, you can see that both of these two FAH null pigs were able to survive off NTBC. And importantly, you can also see that the HT1 phenotype was essentially completely rescued, with significant reductions in total bilirubin, ammonia, tyrosine, and succinyl acetone. And so this data together suggests that wild-type cells had essentially continued to grow and expand in these animals to the point that they had repopulated their liver sufficiently to cure the pigs of HT1. And indeed, when you look at histology that's taken pre-cell transplant, as well as at a period 12 months post-cell transplant, you can see that there's this significant repopulation of the individual liver lobules with FAH-positive pig hepatocytes. So we can use this knowledge as the basis for being able to expand hepatocytes in vivo, and we can then take things another step further and generate 2 additional mutations in these animals, and that's to knock out their immune system. So if we do targeted knockout of RAG2, which stands for recombination activation gene 2, that prevents random assembly of the variable, the diversity, and joining gene segments, also known as VDJ recombination. And that effectively prevents the ability of the animal to produce immunoglobulins and essentially renders the animals depleted of B and T cell function. And then you can simultaneously knock out NK cell function by knockout of the interleukin-2 receptor gamma chain, or IL-2R gamma. And so these knockouts of RAG2 and IL-2R gamma are used extensively in many animal models over the past few decades to generate a severe combined immunodeficiency, or a SCID phenotype. And importantly, and this is really key to the development of our model, if an animal with the previously described FAH knockout were to be coupled to the deficiencies in RAG2 and IL-2R gamma that I just described, you would generate a triple knockout animal known as an FRG animal, and it would, in theory, be able to repopulate livers with xenotransplanted hepatocytes due to its lack of an immune system. So human hepatocytes with a functional copy of FAH would therefore be able to expand in the livers of these animals, as I've illustrated in this diagram on the right. And this has also been done. So going back to 2007, there were these really pioneering studies from the Grompi lab in Oregon that demonstrated that FRG mice could be generated. And in this Nature Biotech manuscript on the left, it was shown that after 16 weeks post-engraftment with human hepatocytes, the mice began producing these really significant levels of human albumin in their serum. And numerous studies have been completed in the meantime using these FRG mice as really these critically important humanized mouse models that can be used for the study of human liver disease. And recently, things have been scaled up by an order of magnitude, with a number of groups having developed an analogous FRG rat model that can also be effectively repopulated with human hepatocytes, as demonstrated in the recent paper on the right here, published in the last month in Scientific Reports, in which rats 7 months post-cell transplant are showing circulating levels of human albumin, in some cases at a really impressive 20 mg per mil, with histology suggesting greater than 80% repopulation with FAH-positive human hepatocytes. So here you have 2 very important rodent FRG models that currently exist and demonstrate as proof of principle that this system for the in vivo expansion of human hepatocytes really is viable. And so building on these important advances, we're attempting to scale things up even further with the development of an FRG pig. So FRG rodents certainly are useful from the standpoint of their small size, their ease of generation. Hepatocytes that are produced in those systems unfortunately face special challenges for their use in clinical applications. And this is why we think that the FRG pig that we're developing has a number of clear advantages for clinical applications over the bioincubation of human hepatocytes in rodents. So from a regulatory standpoint, there's an existing acceptance to the use of pig-derived materials in human therapies. So many of you have likely seen recent reports on the xenotransplantation of pig kidneys and pig hearts into humans. And these experiments are really laying the foundation for our research into the expansion of human liver cells in pigs for clinical purposes. And additionally, and I'll talk about this here in a few minutes, all of our FRG pigs will be clones. And so that should help address any regulatory concerns with genetic drift from natural breeding and further promote consistency in cells derived from these animals. And importantly, scalability is where the FRG pig will really shine. So in the cases of rodents with small livers that are measured on the gram scale, and numbers of hepatocytes isolated from each liver on a magnitude of tens of millions to hundreds of millions of cells, the really large size of the pig would theoretically allow us to use its liver, which weighs over a kilogram, as a bioincubator to produce greater than 30 billion healthy cells, up to maybe an upward bound of 100 billion cells in a single isolation. So that's something that would require hundreds of rodents to even come close to replicating. And then from a production standpoint, pigs are also beneficial due to their well-established processes that have been refined for decades as a result of the use of pigs in food production. So pigs mature extremely quickly to the size of humans. They produce very large litters and have a relatively short gestation period of just under 4 months. And then finally, from a cell purification standpoint, so because human cells will not repopulate the animal's liver completely, we need to take steps to ensure that the human hepatocytes can be isolated with minimal contamination of any residual pig cells that might remain. And so there's obviously concerns here with the presence of xenoantigens and porcine-specific viruses that are being worked out. But we really feel that these issues are further down the road of addressability in pigs than the use of rodent-derived cells, at least in terms of acceptability for clinical applications. And so we're closely following guidance from the FDA and monitoring research that is going to help us mitigate risks that are involved in the clinical use of human hepatocytes that have had both in vivo and ex vivo contact with animal cells. So generating the FRG pig model, we worked in collaboration with Exemplar Genetics out of Iowa in the creation of targeting vectors with the neomycin resistance cassette to develop FAH at exon 5 and incorporating it into AAV-DJ. We infect pig fetal fibroblasts, and then we can expand those cells under G418 selection. And so then resistant clones are identified. We perform similar rounds of AAV targeting to iteratively create knockouts for RAG2 and IL-2R gamma, and that helps us generate the triple knockout pig. And this slide is just simply to show some PCR-based screening that was performed for the purposes of genotyping candidate FRG master fibroblast cell lines. And you can see here that we've identified a particular clone that exhibited the predicted banding patterns for disruption of FAH, IL-2R gamma, and RAG2. So we can then take the master FRG fibroblast cell line and use that in the process of somatic cell nuclear transfer to generate our animals. And I'm sure most of you understand this process, but we essentially source oocytes from healthy female animals. We remove their nucleus, and then through a process of electrical fusion and activation here with our FRG master line, we generate an embryo that contains our desired FRG genotype. And then these embryos are then surgically implanted in surrogate mothers, and then a little over 3.5 months later, the cloned FRG piglets are delivered by C-section. And so we've gone through this process numerous times now with many litters of FRG piglets that have been born in the past year. We've got many more on the way, and this slide is just to give you some brief confirmation that the animals that are being born have our anticipated phenotype. So lack of immune system, as you can see, compared to the lymphocyte populations observed in our wild type animals, You can see that FRG animals are virtually depleted of CD4 T helper cells, cytotoxic CD8 positive T cells, CD3 positive T cells, and CD21 positive B cells. And this is just supporting confirmation to show that our edits are working as expected and that we've developed a SCID pig that lacks an immune system. And furthermore, liver IHC using an antibody specific for FAH shows that our wild-type animals have livers that are chock full of hepatocytes staining positive for FAH, as you might expect. And then in contrast, our FRG animals have virtually no detectable FAH staining, again, providing further confirmation that we've developed a true FRG pig. And so one thing that's really impossible to gloss over here gets back to the fact that these pigs don't have an immune system. With mice and rats, it's fairly straightforward. You can keep these animals in small biobubbles to prevent them from developing infections. That's a little bit more challenging when you're talking about a pig that can weigh over 100 kilograms. So a great deal of our energy has gone into the creation of a barrier environment to help us maintain our delicate FRG animals and really improve their likelihood of survival. And so we've gone to really great lengths to ensure that all of our drinking water is hyperchlorinated and UV treated. Our animal feed is gamma radiated and halo fogged. All of our air is HEPA filtrated to ensure that our FRG pigs are housed in as sterile of an environment as possible. Furthermore, we also limit exposure of animals as much as possible to personnel with all of our animal care techs, our veterinary technicians and our veterinarians. They all have to undergo special testing and gowning procedures before interacting with any of our animals. And this essentially repeats exactly what I just said, but here's a picture of our barrier facility. Again, we really take great pride in the care of our animals, and as such, we're ALAC accredited, USDA approved, and FDA compliant. And again, we go to great lengths to ensure that the pigs are treated properly. So the facility itself is just over 20,000 square feet in size, and it was actually purpose built to house pigs. Though we underwent a significant refurbishment recently to help accommodate the extra requirements that are necessary to house these FRG animals. And so we have a capacity to produce over 300 animals in the barrier environment. And moreover, we maintain a closed recipient high health breeding herd inside the facility with females that serve as our surrogates for embryo transfer as part of that cloning process that I was talking about a little bit ago. And these are just some pictures from the inside showing the cleanliness of the interior of the barrier with a full surgical suite to perform ETs on our surrogate females as well as eventual C-section delivery of FRG animals when they're ready to be born. Again, just some additional pictures of our production area where we house the high health breeding herd, our surgery suite, and recovery area, and then importantly, our FRG production areas where the piglets are initially housed under heat lamps in birthrights with 24/7 care before eventually progressing to sterile growth pens once they reach a certain age. So now that I've shown you some background on our development of the FRG animal, a bit about the challenges that we've had to overcome in order to properly house them, we've begun to implant hepatocytes into our FRG piglets and are starting to begin the process of monitoring the engraftment and expansion of these cells. And because the FRG pigs lack an immune system, we're free to perform both allotransplant of pig hepatocytes or xenotransplant of human hepatocytes using the powerful selection of FAH positivity coupled with cycles of NTBC withdrawal to promote their engraftment and expansion. And to mirror previous experiments that were done using the FAH null single knockout pig, we've started some pilot experiments in which a very small number of wild-type pig hepatocytes were engrafted into our FRG animals. And then after a short time, we're able to see histological confirmation that not only had these cells engrafted, but had likely expanded within some of the individual liver lobules. And again, that's demonstrated by IHC using an FAH antibody as shown in the staining here. And serial histological sections really do confirm that despite repopulation near the edge of this particular liver, the staining likely was not the result of any edge effects, but it likely demonstrates true engraftment and expansion of wild-type pig cells within the bioincubator. And we're also just now starting to see some data coming online where our techniques of administration of primary human hepatocytes into the FRG model are demonstrating clear evidence of engraftment, as you can see here, again with IHC staining against FAH, and in this case showing engrafted FAH-positive human hepatocytes present within the surrounding FAH null parenchyma of the FRG pig. So, in summary, we're still in early but exciting stages in the development of our model. We believe that we're well on our path toward the use of this new large animal bioincubation system to help us produce high quality and consistently available hepatocytes, again, for multiple important purposes. And just to summarize what I've talked to you about today, we've demonstrated that there's been really key progress on the path towards our development of this large animal FRG model. Our FRG pigs are viable and durable. We've got multiple litters that have been generated by our cloning process, and the animals kept alive in our barrier environment for 8+ months, despite their completely immunodeficient phenotype. And finally, we're starting to get to the exciting part of our process where we're starting the beginning stages of evidence for engraftment and expansion of FAH-positive hepatocytes in our FRG animals. And so in the end, our mission is clear. We have a goal of helping develop a better treatment option for the almost 5 million people in the United States that are suffering from liver disease. And we believe that our FRG pig model will go a long way to helping alleviate the existing donor organ shortage. So thank you very much for your time and your attention.